Hello everyone, welcome to our channel Mootechie. Today's topic is mitochondrial energy production. On this slide, we see a table outlining the core concepts, the electron transport chain, the proton gradient, and ATP synthase. The visual focuses on providing heavy explanations with just 4 to 5 key points per slide for clarity. We also see a detailed diagram of a mitochondrion. It illustrates the outer membrane, the inner membrane which has folds called cristae, the intermembrane space between them, and the innermost matrix. This structure is famously known as the powerhouse of the cell, and the image clearly highlights its outer and inner membranes. Now let's move on to the big picture overview of mitochondrial energy production. The electron transport chain occurs in the inner membrane, while ATP is synthesized in the matrix. The primary fuels are NADH and FADH₂, which are derived from the TCA cycle and beta oxidation. Oxygen, or O2, acts as the final electron acceptor, reducing to form water H2O. The entire process is driven by chemoosmotic coupling, where the ETC builds a proton gradient that powers the ATP synthase rotor. Following this, let's get an overview of the electron transport chain itself, focusing on complexes 1 through 4 and the mobile carriers. The diagram shows complex 1, 2, 3, and 4 embedded in the mitochondrial membrane, with the intermembrane space highlighted. The text explains the electron flow. Electrons travel from complexes 1 or 2 to ubiquinone or Q, then to complex 3, then to cytochrome C, and finally to complex 4. Regarding proton pumping, the complexes pump hydrogen ions, H+, out into the intermembrane space. Complex 1 pumps 4, complex 3 pumps 4, and complex 4 pumps 2. This activity creates the proton motive force, represented as delta P, which combines a chemical gradient, delta pH, and an electrical gradient, delta V. All of this occurs in the inner mitochondrial membrane, which is crucially impermeable to ions. Let us now look at the first entry point into this chain, complex 1, also known as NADH dehydrogenase. The diagram shows the complex spanning the membrane. On the matrix side, we have the NADH binding site. Electrons from NADH flow through a path involving an FMN molecule and iron-sulfur clusters. This energy is used to reduce ubiquinone or Q and importantly to pump 4 hydrogen ions across the membrane into the intermembrane space. Next, we explore an alternative entry pathway, from complex 2 to Q to complex 3. This slide highlights that complex 2, or succinate dehydrogenase, accepts FADH₂. A key difference is that, unlike complex 1, it does not pump protons, which means it contributes less to the overall proton gradient. The mobile carrier here is ubiquinone or Q, a lipid-soluble molecule that collects electrons from both complex 1 and 2 and shuttles them to complex 3. Complex 3 then uses a clever mechanism called the Q cycle to transfer these electrons while efficiently pumping 4 hydrogen ions into the intermembrane space. Finally, electrons are passed one by one to a small mobile protein called cytochrome C, which ferries them to complex 4. Coming up, we'll discuss the final step, complex 4 and proton gradient formation. The diagram depicts cytochrome C delivering an electron to complex 4. This complex, cytochrome oxidase, holds an oxygen molecule at its reduction site. Here, a critical reaction occurs. O2 plus 4 electrons plus 4 H plus from the matrix yields 2 molecules of water H2O. For every 2 electrons that pass through, complex 4 pumps 2 additional hydrogen ions across the membrane, further intensifying the gradient. By consuming these chemical protons from the matrix to make water, the pH difference across the membrane increases. The result is a powerful electrochemical gradient, the proton motive force, now ready to drive ATP synthase. Moving forward, let's understand ATP synthase and its process of rotary catalysis. It has a dual component structure, the membrane-embedded F0 unit, which is a proton channel, and the matrix-facing F1 unit, which is the catalytic part. This structure acts as a rotary motor. The flow of hydrogen ions back through the F0 unit spins a C ring and a central gamma shaft, much like a turbine inside the F1 head. This rotation forces the beta subunits through a three-state binding change mechanism: open to bind or release ADP and phosphate, loose to hold them, and tight to synthesize ATP. The proton cost for this is approximately 3 H-pass to rotate the motor for 1 ATP plus 1 more H plus to import the phosphate, totaling about 4 hydrogen ions molecules per ATP produced. As we continue, here is a visual summary tracing the path from the electron transport chain to ATP. The diagram shows all the complexes lined up in the inner mitochondrial membrane. The key steps are outlined. First, electron flow travels from complexes 1 or 2 to ubiquinone to complex 3 to cytochrome C to complex 4, ending when oxygen is reduced to water. Second, the energy from this electron flow is used to pump hydrogen ions into the intermembrane space via complexes 1, 3, and 4. Third, this creates a strong electrochemical gradient, the proton motive force, now storing potential energy. Fourth, and finally, through chemoosmosis, ATP synthase harnesses the backflow of these protons to phosphorylate ADP into ATP. So, what's the bottom line? Let's talk about the energy yield and ATP accounting. For each NADH molecule that enters at complex 1, about 10 protons are pumped, yielding approximately 2.5 ATP. For each FADH₂ molecule that enters at complex 2, about 6 protons are pumped, yielding approximately 1.5 ATP. The difference exists because FADH₂ bypasses the proton pumping of complex 1. Fewer protons pumped means a smaller gradient and less ATP. Remember, it costs about 4 ATP to make 1. In total, oxidative phosphorylation via the ETC produces 26 to 32 ATP, far more than the mere 4 produced directly from glycolysis and the TCA cycle. This brings the total cellular yield per molecule of glucose under aerobic conditions to between 30 to 32 ATP. Thank you for watching. We hope you enjoyed learning about these essential metabolic pathways. Thank you. See you in the next class. Author Mootechie.